네, 감사합니다. Thank you so much for the introduction. Hi, I'm Gina Ko, a postdoc at MGH and Harvard, working at the Ralph Weisslater Lab. And today I'm going to talk about one of the most recently published articles on ultra-fast cycling for multiplex cellular imaging. I'm interested in profiling tumor immune microenvironment time for immunotherapy. Immunotherapy has been highly successful in a subset of patients that were previously incurable, but the remainder do not respond to the therapy at all. So we want to understand this bifurcated outcome and be able to improve the immunotherapy. And it has been found that time has a significant role in determining immunotherapy, so we are interested in profiling them during the course of the treatment. So my postdoc work has been built around profiling time. And then the first part that I'm going to talk about is the ultra-fast cycling for cellular imaging. And then the second part will going to be immunodroplet digital PCR, where we can identify individual exosomes or extracellular vesicles for analysis. So there are multiple methods that we can use to profile time. One can be biopsy, where you take the tissue from the diseased area or the tumor to get molecular information. But this is very invasive, so you cannot really do repeat sampling. Other method is liquid biopsy, where you look at blood-based biomarkers, which is suitable for repeat sampling. But then the issue is that biomarkers are very dilute in the presence of huge backgrounds. Another method is called FNA, fine needle aspiration, where you take small needle into the tumor site and take few cells out of it. This is very suitable for repeat sampling because it's minimally invasive, and also it can get direct information from the tumor. So we're interested in using FNA samples, but they're very scant, meaning that when we take needle pass, we only get few thousand cells out. And flow cytometry, which is a widely available method for single-cell profiling, cannot be used with FNA samples because at least few 100,000 cells are needed to start with. But when we are profiling immune environment, there are so many markers that we want to profile. This is the challenge of using FNA samples. And there have been some current cycling methods where people are trying to profile different markers from the same cells, which means that they are doing staining of the same cells and then destain them and cycle that through so that they can profile multiple markers from the same cells. However, their destaining methods are very harsh because they're using hydrogen peroxide or NaOH to strip off the fluorophore, and also some use barcoding strategy, but it has residual background issue. And also one challenge is that all these methods take at least 30 to 45 minutes for destaining, meaning that if we want to cycle 10 different cycles, we need to wait for 5 hours just for destaining methods. And more importantly, since they are very harsh conditions, it works quite well with the FFPE tissue samples, but not work well with single cells because they don't have any scaffold, they're more fragile. So our goal is to come up with a gentle, ultra-fast method for single cell profiling. So what we want to achieve is to stain the cells and then quench them, and then stain them again with different markers, different channels, and then quench them again. But what we want to do is to quench them at this speed, which is only 1 second. And in order to achieve that, we are using ultra-fast click chemistry, TCO, transcyclooctane, and TZ tetrazine chemistry, so that the fluorophore can be quenched by the quencher instantaneously. So here's a little bit of chemistry, but just to briefly go over, we make TCO with PAC4 for good solubility, and then we conjugate that with fluorophore, which NHS or TFP, so that we can react those with the amine group of the antibody, so that the antibody can be conjugated with the fluorophore and TCO. And then the other part of chemistry is that we're making tetrazine quenchers. And for quenchers, we're using BHQ, black hole quencher 3, which is very widely used quencher also in qPCR. and then you conjugate with tetrazine so that this TZ and TCO can click, and then the quencher can be either by contact or FRET right next to the TCO fluorophore so that we can achieve quenching at very high speed. We first look at labeling efficiency because whenever you modify the antibody, you want to make sure that the antibody is working pretty well. So what we first did is we looked at different equivalents of fluorophore TCO, and then we compared that to commercially available fluorescent antibodies, and then showed that both of them, the brightness are comparable to each other. And then we looked at quenching efficiency to make sure how well our method can quench the cells. For Alexafluor 647, we first start with 100%, and it can go down to 1% for the residual intensity, so we can get more than 99% quenching, and all different dyes that we tested were able to get at least more than 95% quenching, which was extremely high. And you can actually see them in the images where you can see after staining before quenching, and then this is after quenching where you literally do not see any signal from the cells. But our ultimate goal is to achieve ultra-fast quenching. So in order to test it out, we looked at different time points of the quenching time from 5 minutes, 1 minute, 30 seconds, and 10 seconds, and see how fast our quenching can work. And you can see that at only for 10 seconds, we were able to see all the stain signal of the red disappears within 10 seconds. And then the reason that I brought it down to 10 seconds was because whenever I was done with the pipetting, it was already 10 seconds. So in order to better characterize the quenching kinetics, we used the fluorimeter for measurements. So here we were able to find that the half-life was less than 0.5 seconds, which was extremely fast. And that was achieved because of the huge rate acceleration up to a few thousand times. And we're expecting this is because multiple pair of the click reaction happen at the same time, and somehow they can have cooperative effect so that it can accelerate the quenching rates. And we were able to observe that rate acceleration for all different fluorophores, even though the values were slightly different from each other. And then we were able to show that we can image multiple targets in the same cells. So you can see that this is phospho-S6, EGFR, phospho-6 from the same cells, and also different channels. And for all of them, staining and quenching, you're able to get really clean signal, no signal left over from the previous cycle. And using that, our final application was to apply to tumor FNA samples and try to profile their immune cell composition. So here we looked at 12 different markers plus DAPI, and then did 4 different cycles, and then we were able to achieve intratumoral immune composition of looking at 8 different cell types with different markers. And then eventually our goal is to be able to profile immune microenvironment during the course of the treatment so that we can link that information to response to therapy to improve the therapeutic efficacy. So for future directions of this study is to do multiplexed patient-derived live cell imaging. Patient-derived is a huge advantage for our methods because we don't really need to modify the gene in advance to express YFP or RFP because we can always label them afterwards using antibody. And also since our method is very gentle, it doesn't damage any cells, it can be used for live cell imaging. So currently we're testing on live cells to see if there's any good application for this method. And then the second half of my talk, we're going to be working on immune droplet digital PCR. So we are trying to answer the same question, how to profile time Eevee for immunotherapy. And there's a great biomarker called Eevee, extracellular vesicles, that include microvesicles and exosomes that are shed from their mother cells. And they have rich molecular information of their mother cells, including DNA, RNA, and proteins, which can be a great marker that are found in the blood, so we can easily identify them in the blood. But then the issue of time Eevee profiling is we have to fight against huge backgrounds. every single cell, all different cell types secrete these EVs, and then in the blood, there are going to be millions of blood cells that are secreting their own EVs, and then the EVs that we're interested in are coming from tiny tumor in the body. So in order to find that rare population, our method is to profile single EV, because our problem to solve is to find a needle in a haystack, and in order to do so, we're going to make small containers and put each hay or needle into the container and only pick up the containers that can have the needle in there. And then the container that we're using are droplets that we were able to make using droplet microfluidics. And then I first worked on Droplet digital PCR system, and I'm currently scaling up using sequencing technology. So here's the single Eevee Droplet digital PCR pipeline, where you first start with Eevee coming from cells, and then you label the Eevee with antibody DNA conjugates. And this DNA has two different purposes. One is to be a template for barcoding, because I want to profile different proteins, which means that I want to target different antibodies. And then we were able to distinguish that using the barcode from the DNA. And also the DNA can be used as a template for amplification. If you think about EVs, they're very tiny, nanovesicles, 100 nanometer up to 1 micron. So the content inside is very rare and scarce. So we really need amplification so that we can get readout from single EV level. So we encapsulate it in droplets, amplify using thermal cycling, and then the EVs that contain the target will go into fluoresce. So here you can see the Droplet digital generator, where I'm going to play here. So you can see the microfluidics where we have two different input channels. One master mixer is coming through, and then the other one Eevee is coming through. But since Eevees are very small to be visualized, I'm using 4 micron beads for visualization. And then you can see that the water is pinched by oil to make droplets that have single Eevee and master mix in there. And then we were able to show that by using Poisson's distribution, statistically, we can encapsulate one Eevee or bead per droplet so that we can do single Eevee analysis. And then we first did titration comparison by comparing the number that we counted using our system to the number that we counted using NTA, nanoparticle tracking analysis, just to make sure that we can accurately count the number of EVs that we're interested in. And we were able to find that our dynamic range was 3 orders of magnitude, and then our sensitivity matched to other systems. And we also did control experiment using the markers that we already know. So we looked at U87 parental Eevee, which has low expression of EGFR, and U87 wild type Eevee, which has high expression of EGFR. And we were able to quantify that by counting the number of EGFR positive droplets. And we also did some specificity tests by doing competition inhibition assay, where we labeled the Eevee with pure anti-EGFR antibody first to block the antigen site, and then show that we capture less and less, or we detect less because of the specificity. And then we were able to show that we can do multiplexing. So rather than just identifying single EV groups, we wanted to identify double positive or triple positive subgroups that can be meaningful for diagnosis. So here we looked at EGFR and EpCAM, and using that, we were able to create a plot similar to flow cytometry plot, where you can see double positive populations and single positive and double negative population. And we also increased that to 3-plexing so that we can look at different subgroups of the EV from the same sample. So our final application was to profile immune population at single EV level. So here's the cell expression of different cancer cell lines, and then EVs that are derived from the cells. And you can see that they are very highly correlated to each other. This is why we can actually use EVs as a substitute marker for cells, because tumors are usually in hard-to-reach area, but then EVs coming from the tumor can be easily found in the blood, which can be a great biomarker for that. And then we profile PDN1 expression because this is a predictive marker for immunotherapy for different cancer cell lines. So currently, we're also working on measuring during the course of the treatment so that we can see how the level changes over time. But also at the same time, I'm a bioengineer by training, so I'm very interested in technology development. So I'm currently scaling up to be able to profile millions of EVs and hundreds of markers by using sequencing technology. So the single Eevee protein sequencing technology pipeline is as follows, where you start with EVs, label with antibody DNA conjugate, and then encapsulate them in droplets with barcoded bits, and then make sequencing library. But here, we cannot use single-cell sequencing protocol that's already out there, because when you think about the scale, EVs are 100 times smaller than a cell, meaning that their content can be a million times less than what one cell has, which means that we need a new ultra-high sensitive protocol so that we can get readout from the single Eevee. So I've been spending a lot of time to work on the protocol to read out single Eevee rather than a single cell. So there are two different components of our platform. One is barcoded bits, where we have barcodes that we make using split and pull approach and UMI, unique molecular identifier, so that we can actually count the number of molecules. And then A is hybridization region, which we're going to bind to the antibody DNA conjugate. And here's how we make the bits. We do extension using the barcodes, and then we split them, dehybridize so that we can get a single barcode, and then repeat the steps so that we can increase the diversity of the bits. And the second component is antibody DNA conjugate. The A region that I showed you before, we're going to bind here, A prime or A star. And then here's the antibody barcode where we can know which antibodies the protein was targeted. And then we have T7 promoter sequence, which is used for in vitro transcription, which I'm going to talk about later. And then we developed our own way to make antibody DNA conjugate using TC/OTC click chemistry, which was more efficient, and we don't need washing step in the end. And also it was way faster than other chemistry. So here's the protocol that I developed. The first step is extension in droplets. We have barcoded bits and antibody DNA conjugate that capture Eevee, and then the extension will going to happen so that we can have a single strand that has all the information in there, meaning that we know which Eevee the protein is coming from, and then how many protein molecules we have, and also what protein we are targeting. And then the second step is in vitro transcription, meaning that from DNA, we are generating a lot of RNAs from DNA. And we are using IVT for two different purposes. One, in order to amplify the signal as much as possible. Two, we want to get rid of the DNA that didn't go proper extension, which can be a potential cause of crosstalk in the future. So after IVT, the amplification, we use DNAs to get rid of all the potential cause of DNA, and then do purification of the RNA, and then do RT to convert to DNA, and then PCR, and it's ready for sequencing. So here's the single Eevee sequencing droplet generator, where you can see in this video. So here we have closely packed bits that are hydrogel bits, so they're pretty flexible. So we can closely pack them, and then this channel length is actually, width is actually smaller than the hydrogel bits, so they're squeezed, and then only one can come out at a time, so that you can see one coming out, and then we have some master mix and Eevee solution. They are all going into the droplet so that the reaction can happen in droplets. So we first checked our amplicon to make sure that we were able to make the amplicon that we wanted. So we do qPCR, and then for positive control, we use bulk Eevee that we prepared in a tube. And for bulk and also our single Eevee using droplets, we were able to achieve the size that we wanted. And then we further checked the actual sequences of the amplicon that we made using Sanger sequencing, which matched really well to the template sequence that we were expecting. And then moving forward, we wanted to check crosstalk just to make sure what percent of the information actually correctly aligns to the reads that we want to profile. So in order to do the testing, we looked at 2 different Eevees, labeled them with the same antibody, but different barcodes separately, and then mixed them together. And then we ran our protocol and then the sequencing, and we were looking for the reads that align to both barcode 1 and barcode 2, which will be wrong and considered as a crosstalk. And we were able to find that the crosstalk percentage was pretty low as 5%, so we were able to move on and do actual pilot Eevee profiling. So here we just looked at a few thousand Eevees and then 2 different markers. These are double positive Eevee subgroups, and we were able to profile individual Eevee at single Eevee level for different markers. So right now we're ready to move on and actually look at the markers that we're interested in coming from tumor and coming from the host immune cells, and try to look at millions of Eevees and hoping to be able to find very rare population that can be indicative of the response to therapy and for monitoring. So with that, I want to acknowledge my postdoc advisor, Ralph Weiseter at MGH and Harvard, and my chemist advisor, Jonathan Carlson, and Julie, and David Weiss, my collaborator working on Droplet stuff at Harvard University with Yongchen Wang, and everyone from the Weiseter Lab, MGH, Harvard, and VIS, and my postdoc fellowship, Schmidt Science Fellows, for all the support. Thank you so much for listening to my talk.